पहले तुम्हें कॉपी करना होगा ना।
morning.
Good morning. Good morning, everyone. Sarla from Unisem. Good morning, sir. Good morning, Aparna. Yeah.
Hi, Joseph.
Hello, Umesh. Good morning.
Good morning.
Okay. Good morning. Sorry, there was some problem with the network. It took me out and I am back again. Joseph, how are you? Good to see you. Can you hear me?
Yes, I can.
Oh, okay. Good, good. You
Hello.
yeah, please start. Thank you, Aparna. And thank you, Joseph, for accepting today's invitation to talk to us. It's my privilege to introduce Dr. Jozsef Mozsar to the listeners. He's a seasoned plant breeder with over 28 years of experience in vegetable crop improvement with a distinct specialization in tomato breeding. Renowned for his mastery of breeding strategies, genetics, and crop adaptation, he has successfully pioneered innovating tomato varieties with superior agronomic traits tailored for both open field cultivation and protected environments. That is a unique combination. Not many have both, either open field or protected, so that's very nice. As tomato breeding team lead in Syngenta, he played a pivotal role in guiding breeding teams across Europe, Asia, and North America, fostering the development of high-performing tomato varieties to meet diverse marketing demands. Prior to working with tomatoes, he also contributed to the cauliflower breeding and conducted research on tissue culture and genetic transformation. Grapevine. Sorry, can you hear me?
Yes.
Armed with PhD in biotechnology, he is deeply committed to integrating traditional and modern breeding methods, which I'm sure we will hear a lot today, to drive crop performance, sustainability, and global food security. Today, as an independent consultant, he supports breeding programs of companies and institutes, helping to shape the future of vegetable business through scientific innovation and collaborative excellence. With that, the floor is yours, Joseph.
Thank you, Surinder. Thank you for the introduction. And let me start to welcome everyone in this call and express my honored feeling to be here and to be invited to hold this presentation. With that, I would start.
Sharing your slides.
Yes, I hope I will select the right window.
Yeah.
Let me know when you see my presentation.
Yeah, it's come. Yeah, it's there.
Okay.
Yeah, we can see it.
Okay. Thank you. Thank you very much. Okay, so let's start with the presentation. I've been asked to talk about tomato breeding and especially within the tomato breeding, talk about disease-resistant breeding. What is resistant breeding? The definition, what I found and like is very simple and very straightforward. So resistant breeding is a process of developing plant varieties with enhanced ability to withstand disease and pests through genetic improvements. The importance of it is obviously reduce reliance on chemical and pesticides in crop growing, enhance the crop yield and stability, and of course, support the environmental sustainability, just having less need of using chemicals and pesticides. And with this in this way, it supports the global food security, which is getting more and more on focus. What is the goal of vegetable breeding? It's also very simple, to breed competitive varieties. And what is the simplest and very short definition of competitive varieties? The variety what meets the needs of farmer, trader, and consumers. Let's see the need of these three groups. If you look at the growers and traders, their interest is to maximize profit. And if we see the consumer's interest, it's rather maximize the satisfaction. How the resistance contribute to their interest and contribute to make your variety a successful variety. For the growers, of course, the resistance means lower cost because, as we discussed, as I said before, if you have resistant variety, you are less dependent on chemicals, so you need to use less chemicals, which is basically less cost. Of course, indirectly it contributes the yield stability and the overall yield, but the primary interest of the resistance is from the grower is the less input cost. If you look at the trader's interest with resistant variety, they can rely more on the growers, so they can be more confident to having the supply. And the less use of chemicals, also contribute to the food safety, which is also interest of the traders. If you look at the consumers, the number two interest and let's say benefit through the resistant varieties is the food safety and the environmental aspects, which both are getting more and more important from a consumer point of view all over the world. If you look at the type of resistance, we may classify as a vertical resistance and horizontal resistance. Vertical resistance is controlled usually over one or a few major genes and provides significant protection against specific pests and pathogens. While the horizontal resistance usually controlled multiple genes and provide partial resistance, but for a broader range of pathogens and pests. And usually, while the vertical resistance can easier overcome by the pathogens, the horizontal resistance is more durable. The other classification we can split the type of resistance is a high resistance, which significantly reduce the prevent the growth of reproduction and impact of the pest or pathogens. And the intermediate resistance refers to a lower level of resistance, which anyway provide better ability to the plants to tolerate a certain level of infection and damage caused by pests and pathogens. Compared to a susceptible variety, but this level is lower than the high resistance. Plants with intermediate resistance may show some symptoms and damage, but they maintain higher level of productivity compared to the susceptible variety. Another aspect we need to investigate when we are talking about resistance breeding, the role of genetic diversity helping the resistance breeding. If you have appropriate level of diversity in your germ plants, it increase or provide wider range of availability of resistant genes for your breeding program. Enhance adaptability to the crop to different environment conditions and disease pressure. Also reduce the likelihood of resistance breakdown due to the pathogen evolution and facilitate development of new varieties that combined resistant trait. Also ensure long-term sustainability of resistance breeding strategy. Overview of tomato breeding in general. Tomato is the most widely cultivated vegetable crop globally in terms of production volume. Breeding efforts for tomato usually focus on improving yield, fruit quality, and resistance to biotic and abiotic stresses. And tomato is a leading crop as the most important vegetable crop always. Of course, the crop which has a lot of efforts from the breeding community. So traditional and modern breeding methods are used for tomato. The importance of resistance breeding, crop losses due to the disease and pest are the major challenge of production in tomato. Develop resistant varieties, of course, as we discussed previously, reduce the need of chemical pesticides, making farmer more sustainable and cost efficient. And the resistance breeding also has contribution to increase food security and ensure tomato production under various environmental conditions. Tomato itself, a diploid plant with 12 chromosomes as a haploid chromosome set, cultivated in diverse climatic conditions. If we consider the protected growing, let's say it's the range of the geographic zones overspan from the tropical to the from the equatorial to the almost the Nordic conditions. The genetic center of tomato is the Andes area, but the domestication occurred in Central America and Mexico. The flower structure promotes self-pollination, so that's why the hybrid selection is done through manual emasculation. Just to support further the importance of tomato within the vegetable production, you can see that territory-wise is the second, followed the onion. It's 18% of the surface, what is globally used for vegetable growing, used by tomato. But if you see the volume of the production, it is number one. If you think of the productivity of protected crops, it explains why territory-wise tomato is just the second, why in production volume it's far the number one. If we look at the tomato seed market, the estimated market in 2022 was $1.2 billion. Shortly, the abiotic stress, we are not going to enter into this field, but just let you know the challenges what tomato growing face from abiotic stresses. Here we have temperature, heat stress, cold stress. It is due to the wide geographical area where tomato is grown. The soil, of course, can be also the source of the stress, having high salt content or not enough water, and also the growing application, the growing condition can also may have some challenges to the tomato plants or tomato varieties, such as growing with hormone application. It's probably less typical in India, but in Asia, Southeast Asia and South Asia, but more common in protected crops. Oops. And here are the list of the biotic stress. So you can see the list is extremely long. So tomato face a high number of pathogens, which also makes the tomato breeders' life quite difficult. We'll see that luckily there are quite wide range of resistant genes available to tackle these biotic stresses. So we have a problem with viruses, with bacteria, even phytoplasma, also pests, and the longest list is the fungi diseases. Where we can have resistance against these stresses, the most widely used source for resistance is a viral relative of tomato, domesticated tomato. For some disease, you may find resistance in the domesticated tomato germplasm. Also, mutagenesis can be the source of new resistance, while transgenic approach, I do with all the controversy discussion acceptance around it, makes it less easy worldwide, depending, of course, the market acceptance. And recently, the new technology, the gene editing, also enable researchers to design and create redesigned genes, creating resistance against certain pathogens. For the first step of using new resistance is trait discovery, where we need to go through a kind of quite strict process to start a trait discovery project. The number one step is problem identification and trait prioritization. I will talk a little bit more in details later on. The second step is general germplasm exploration and the general assessment of genetic variation. So the genetic mapping and identification of candidate genes. 4 is functional genomics and gene validation. 5, the fifth step is marker development and integration in breeding. This next step is trait deployment and validation through field trials. And the last step is commercialization and variety release. Let's go through this point one by one. When a company decides it, I would like to talk about this point from the company perspective or point of view. Before a seed company decide if they start a trait project, trait discovery, first they need to analyze the significance of the disease, which means the impact of the disease in production and indirectly the market value of the trait. Of course, it is an estimation. The other aspect they need to investigate is the required investment in time, in terms of time and money. And having these two information or estimation, they can calculate the return of investment. And of course, before the project starts, they need to assess or try to assess the probability of the success. If this exercise support to start the trait project, then the next step is germplasm exploration and assessment of genetic variation. So where you can find the resistance gene. As we discussed before, the wide relatives are the major sources. Heirloom and landraces are also a valuable asset if you screen for or look for new resistance. In a few cases, you may find the resistance in early germplasm. What happened recently with the TOBRFV, the intermediate level of TOBRFV resistance is available in early germplasm. And of course, you can screen mutant populations which sometimes also provided a resistant source for tomato breeders. The next step is genetic mapping and identify the candidate genes. Here you have several approaches. The QTL mapping is one of it, where you use segregating population F2 real snails to map quantitative trait loci associated with traits. GWAS is widely used for this purpose, associate genetic markers with traits across diverse genotypes. And bulk segregant analysis compare DNA from plant with contrasting phenotype and identify the linked markers for the trait of interest. Also used to find orthodox genes, the comparative genetics, when you have resistance in other species and you try to find conserved genes and regulatory elements in tomato based on the information received from other species. The functional genomics and gene validation. Honestly, this is the step which is rarely used by seed companies. It's rather, let's say, academical part of the academical research. These technologies, the RNA sequencing, through this technology, you can identify differentially expressed genes related to the traits, so confirm that the gene that you find has altered RNA. The proteomics and metabolics study the protein and metabolite profile to link biochemical pathway with the traits, and use mutagenesis and gene editing to validate the candidate gene function. But as I said, it's a rather academical research. Of course, the last one is related to here. Overexpression of knockout studies further gives you the confirmation that may give you the confirmation that the gene that you find has the expected function. Next step is the marker development and breeding integration. It makes the breeder's life much easier if you have molecular marker linked to the trait, because it makes quite easy to follow and track and trace the genes throughout different breeding processes. Here I put SMP and SSR marker, but honestly, recently the SMP markers are what are most widely used for molecular marker purpose, and this enables the marker-assisted selection, which, as I said, makes the breeder's life much easier. The next step is integrate the validated traits loci into elite germ pairs through back crosses, and also the other way where you can use molecular markers is to improve the overall selection efficiency, tracking the desired traits. Trait deployment and field validation. When you have the converted lines, converted hybrids, or newly developed hybrids with new source of resistance, you need to validate the efficiency of the gene. So set up multi-location trials gives you the confidence that the trait stability is there, and it's a very important step through the commercialization, because the variety what will harbor this resistance need to be very stable, stably expressed the resistance in various field conditions. Also, the other variability which makes sense to test the genetic variability in your germplants. So if you convert your materials, it's advisable to convert materials which is representative, which variations representative to the germplants of your breeding program, just to study the efficiency of the gene in diverse genetic background. And the last step is evaluate economic performance and resistance durability and potential trade-off. We will see that the resistance genes usually has some negative impact, especially those which derives from white tomato species. I will talk about that later. And the last step is commercialization of the varieties and variety release. You may create new varieties with using resistant lines newly developed, but also via backcross you can convert existing varieties, existing hybrids. Of course, these converted hybrids and the newly developed hybrids needs to go through the market assessment and variety registration. And in some cases, when the owner, let's say the company, see the potential to put intellectual property protection on the gene, they do it. So if it's applicable, they go through with the patent application on plant variety rights. As I said before, when we have seen the long list of the pathogens which are important in tomato growing, our tomato breeders luckily has a wide range of resistances what they can use. The following tables I summarize the most important ones and their origins, which you can see that usually this resistance comes from white species, which usually has quite a distinct behavior compared to the domesticated tomato. So you can see on the list various white species. Solanum pimpifolium is the closest among these white species to the cultivated tomato. So usually if you have a resistant gene from pimpinellifolium, you have the less negative impact in your germplants, while other distinct varieties like Penelli, Peruvianum, or Chilense, or Haprochites, I will show you a picture later on. They are quite distinct from the Solanum lycopersicum. So integrating resistance from them through conventional breeding also brings flanking region of their genome to the germplants, to the cultivated germplants, and that may cause some problems during the breeding. So these are the genes against bacteria. The list is a little bit less than against fungi and viruses. So this picture shows what we have to think of if we are talking about white tomato species. You can see that these plants, even hard to believe that they have any link to tomato. They live, as I said, the most of these varieties are available in the Andes region, where they live in a very harsh condition. If you let me tell you about a story I always cite when I talk about white tomato. When I started as a cauliflower breeder, and once we visited a grower who had several crops on his field, and among them there was tomato, and the tomato crops looked extremely nice. And we told the guy that, oh, your tomato is really nice, you can be really proud of. And he said, oh, tomato, it's easy to grow, it's a weed. Look at my cauliflower. And okay, we said, the guy is a little bit was exaggerating, but when you see these pictures, you can understand that yes, the white tomato was a weed, and even we cultivated for 1,000 years. It has the domesticated tomato, it still has its robustness. Next step is, next picture shows you the fruit of the Habrochites. So you can see the size is really small, and even in mature stage, it's not red. It's green, a little bit yellowish. So if you bring genes other than the gene of resistance from these white species, you bring a lot of undesired traits into your germ plants, which may cause negative impact on the variety performance. So number one is the weakened vigor. So if you convert your line or hybrids with TY1 or MI, especially in homozygous form, they decrease the vigor of the plants. Consequently, the yield will be lower as well. The pH 2, 3, TY1, MI, and I3 sometimes reduce significantly the fruit size. You also may have the problem of necrosis on the foliage and the stem of the plants. Genes like CF2, I will show you a picture, can cause serious necrosis on the foliage, while TY1 and MI may have yellowing as a consequence of their presence. Fusarium radici, FRL gene, and SW5 also may have negative impact on the germination, and the fruit quality also can be impacted. The genes I listed here, LV, DY1 and DY2, so these are which I found sometimes very negative on the fruit quality. Of course, the extent of this negative impact is strongly depend on the background of your material. So these are the pictures. If you see here, probably
Yes, we can see.
Okay, so can you see the pointer now?
Yeah.
Okay, so on the left hand side picture, you can see the necrosis caused by the CF2 gene in homozygous form. In heterozygous form, this necrosis, let's say, disappears, but under certain condition, you may have the necrosis even in heterozygous form, of course, with less extent. The same with the yellowing. So this is a picture made of homozygous TY1 genes showing the yellowing on the leaves. And yes, on the right hand side, on the lower corner, you can see the scary blossom end of the plant, which harbors the LB genes. Other negative effects like deformation of the leaves and very bad setting may also be the consequence of the presence of resistance genes. Okay, let's see if we have the genes, how we can incorporate into our germplasm. The most classical approach is the selection. So if you have the resistant source, you may cross with your edit materials and try to select the best plant with desirable traits and the best agronomical values and propagate those plants through over generation. A little bit more straightforward is the backcross breeding when you just incorporate the resistance from the donor and try to recover the recipient germplasm, the recipient genome through several backcrosses. The other conventional breeding method where you can utilize the resistance if you cross resistant and susceptible lines to produce resistant hybrid. Of course, this works only if the resistant trait is dominant, partial dominant, or intermediate, has intermediate inheritance. Through the selection, this is the most classical way. I would say this happened before the molecular markers. So you can do the selection at seedling stage, expose seedling to pathogen, then transplant the survivors. The advantage of this approach that you can screen a large number of individuals. The negative or disadvantage that in some cases the seedling resistance doesn't correlate fully with the adult plant resistance. So this you need to be aware of. You also can screen phenotypically at adult plant stage. So you just transplant your plant on the field, exposed to the pathogen, either through inoculation or just providing the condition favorable for the pathogen. The advantage is that of this approach that this is more reliable information about the resistance because you test your plants on the field. Agronomic traits can also be considered contrary to the seedling stage screening. The disadvantage is that it is really resource intensive. Why? You can screen thousands of young plants in a few trays. To grow the plants up and then put in screening requires a large number of large amount of resources. obviously. The other conventional breeding method is the backcross. Here you have the donor with the gene of interest and the elite recipient line, and through the backcross process you try to incorporate the resistance into your elite germ, elite line, and with the less linkage drag possible. This backcross in the past was done by screening. This picture shows a young plant screening, which enabled the breeder to follow the gene of the interest, the resistance. And the hybridization, coursing dominant resistant line and susceptible lines result resistant hybrid. With hybridization, we also may reduce the negative impact of the resistance genes, which is more intensive or more negative when the gene is in homozygous stage. And also, if you have different resistant genes in the lines you are crossing, you can primiding the resistant genes, which may deliver a higher level of resistance at the hybrid level compared to the parental lines. So the good classical example for this approach or for this case is the TY1, TY2 gene combination. Modern breeding techniques, the marker-assisted selection. As I said, having a molecular marker makes the breeder's life much easier and much more efficient, the breeding process. Also, genetic modification is a modern technology, relatively modern breeding techniques, which may deliver resistant varieties, but as I said, the public acceptance for genetic modification is not there yet globally. So there are areas like Europe where this is strongly opposed by the public opinion leaders and, let's say, the people. Genome editing is a relatively new technology compared to the mass and genetic modification. So with this technology, you can precisely modify the gene to enhance the resistance and minimizing the unintended genetic change. This is a very sophisticated tool, so you need to understand very well the gene, and you need to know where you need to modify the DNA of the gene. Also, I added here the genomic selection uses genome-wide marker and predictive model to select the individuals of desired hybrid with improved resistance. This, I would say, the newest technology, largely used in field crop breeding, but as the technology evolves and becomes more affordable, more and more vegetable breeding program use genomic selection for this purpose. Okay, let's go into the details for the market-assisted selection. You can do the selection at seedling stage just in case of the phenotypic selection. The advantage of this approach that, as said before, large number of individuals can be screened, and after the screening, you discard susceptible plants so you transplant or continue with resistant plants further. Also, you can do the marker-assisted selection, testing the adult plants on the field for the gene of interest. The advantage of this approach that agronomic traits can be considered when you make your final selection, but once again, as it has discussed before, it requires a large number of plants on the field, which is resource intensive. These are the diseases we have reliable markers. To my knowledge, of course, there are some markers which has not been revealed, which has been developed by companies, commercial seed companies, and they use for their own purpose. But definitely the development of the molecular markers is very rapid. So this list is growing, let's say, every day. How we can use molecular markers with backcross? With the classical backcross, you just trace the desired traits from the donor, so you follow with molecular marker only the traits what you want to incorporate from the donor to your recipient material. But more and more the backcross with background selection is used, which accelerate the process as actively select and follow for the recurrent genome using more marker which covers the whole genome of the tomato. This is a scheme of the classical backcross. You have the recipient, you have the donor. There's red circles shows the gene of the interest we want to incorporate. So if you follow this gene with molecular marker and you go through several cycles of backcrosses, with each backcross you increase or recover the recipient genome, and after a number of backcross, which here is shown BCN, but usually 4, 5, 6, you have a very good recovery of the recipient marker. And if you followed the donor gene, in our case it's the resistant gene with marker, you may have it incorporated into the elite germ plant, the recipient germ plants. Okay, so this is you don't need you can do the selection in young plant stage, so it also reduces the resource what is required. This slide shows the kind of distribution of the recipient parent genome in BC1 population. So the average is 75. The standard deviation is 5 to 5.5. And if you have 200 individuals, you may have a maximum recipient genome around 88%. So it means that if you have this BC1 population and you can actively screen for the recipient or follow the recipient genome, you can identify the percentage or the proportion of the recipient genome in each individual. And if you have this information, you may select those on the right-hand side of the diagram to follow, to continue and use those plants or one of those plants in the next cycle of the backcross. So this is the classical foreground selection when only targeted locus is followed by marker. Sometimes they use, but it's not very common, a flanking marker around the chromosome part where the gene of interest is located, to see that here the recipient genome is restored. So with this approach, you can minimize or reduce the flanking or region from the donor plant. So let's ensure that you may have less linkage drag at the end of the process. And the third approach, what just explained you, the background selection. So you follow the gene of interest with one marker and with several markers, which covers well through the whole genome. You follow the recipient genome, increasing the restoration of the recipient genome overall. So this diagram shows the evolution of the restoration. The yellow line shows the classical foreground selection when only the gene of interest is followed by marker. The yellow shows the green shows the flanking approach when you use two markers, and the red one is the target and background selection. So you can see that the restoration of the recipient background in this approach is significantly better, faster. So in BC3, you may have a very good recovery of the recipient genome. So by this way, you save one or sometimes two or three back cross cycles. Of course, this approach requires more use of more marker, which make the process more expensive and more complex. Okay. What are the challenges, to summarize, what are the challenges in resistant breeding? The genetic trade-off, as we have learned, the resistant genes can sometimes lead to reduced fruit quality, yield, fitness, or other agricultural traits due to the undesired genes transferred from white species together with the resistant genes. Evolution of the pathogen is constant, so pest and pathogen can overcome resistant genes, which requires continuous breeding. The intensity of this overcoming and evolution varies pest by pest or pathogen by pathogens. The complex genetic interactions, sometimes resistant genes are controlled by multiplied genes, which makes breeder's life more complicated, and the process of delivering resistant varieties more time consuming. There are regulatory and market acceptance issues. Transgenic and genome editing crops face regulatory hurdles and public perception challenges. Sometimes we face limited genetic resources where we don't have right resistant genes, or we haven't found it yet. The environmental variability, climate change, and shifting pathogen population can alter the disease pressure. This is what we are experiencing now, and this brings new challenge to the resistance breeding as pathogens and pests appears in area where they didn't exist before. Developing resistant varieties is costly and takes several years. Of course, it's obvious. And trade discovery is not affordable for all players. So especially small size companies or private breeders, they don't have the resource to run trade discovery projects. The future prospects and innovation in the field of the resistance breeding. Advances in genomic sequences enables us to rapid identification of resistant genes via whole genome sequencing and genome-wide associated studies. The role of artificial intelligence and big data in plant breeding with predictive models using big data and artificial intelligence help us to optimize the selection and analyze the complex genetic interaction. This is important, especially if you have a complex inheritance of the resistance. Gene stacking approach, combining multiple resistance genes, pyramiding to create durable and broad spectrum resistance. There are examples, as I showed before with TY, stacking TY genes or pH genes against late blight. But yeah, this is a way which we need to put more efforts because it seems that the single gene in some cases is not a long-term solution for keeping the resistance alive. Precision breeding technologies such as genome editing, it's a very advanced technology and would help the breeders to develop, or scientists develop, new resistances without too much altering the agronomical traits in your plants. Integration of resistance with high yield and quality traits, developing varieties with balanced resistance in market, consumers, and preference. It's a challenge because, yeah, as we discussed, sometimes the resistant genes, or very often the resistant genes, have negative impact on the agronomical traits. And the climate resilience breeding, this is what in the case of resistant breeding, we need to consider more and more, because the level of resistance very often is impacted by the abiotic stresses such as heat, for example, in case of the nematode resistances. So when the soil temperature is high, the MI gene efficiency is decreasing dramatically. The key takeaway of this presentation, very simple: the resistance breeding is essential for sustainable tomato production and food security. It's very clear that we cannot stop with resistance breeding. Integration of conventional and modern breeding techniques has led to significant advancement in developing disease-resistant varieties, especially artificial intelligence, big data, genome editing. Yeah, these are the techniques which may help the breeder significantly to be more successful and more efficient in resistant breeding. And genetic diversity, advanced breeding technologies, and continuous research are critical for overcoming challenges and improving resistant durability. The future direction, enhancing collaboration between breeders, researchers, and farmers. I believe it will be more and more important, as the problem that breeders and researchers has to tackle is getting more complex. The knowledge and the expertise what is accumulated in certain groups cannot be enough to tackle the problem. So collaborating between these teams may increase the efficiency and increase the probability of the success against new challenges. Utilizing advanced tools such as genome editing or genomic selection to refine the resistant trait without compromising yield and quality. Expanding breeding efforts to incorporate resistance to emerging disease and climate-related stress. This had been discussed before. Call to action. I would deliver that message that support initiative and policies to promote sustainable resistant breeding, and once again encourage the knowledge sharing and technology transfer to improve tomato resistance worldwide. Thank you for your attention.
Thank you, Joseph. Very nicely summarizing a whole lot of things that have happened in the tomato world from the sources where we got the diseases, the summarized slides on what is available and what are the markers and what are the inheritances of that, and where we should go in the future. Before I ask if I had a few things that I want to say, but I think let us first hear from the people in the queue. Anybody who wants to speak and ask a question directly? Can you moderate that? Yes. Yes, Nandish.
Hello, sir. Can you hear me?
Yeah, please.
Yeah. Nice presentation, sir.
Thank you.
I wanted to ask one specific question. So you mentioned that from Solanum pimpernifolium, we have used a lot many resistant genes because it is a relatively close wild relative compared to other species. Is there any bridging species that we can use to generate population from Solanum chilense?
Sorry, I didn't get to the last sentence what you said.
He's
So
saying
I just hear the chilense.
Yeah,
I'm working with Solanum chilense. So is there any bridging species I can use to develop population?
Yes, you can use. Honestly, I have a slide from my previous past presentation which shows this kind of interaction between the white species and the domesticated tomatoes, which one can be crossed directly. Chilense is definitely a wild relative, not very close to Solanum lycopersicum. I don't know by heart. I don't want to say any stupid things, but if I remember well, with Chilense, you can develop interspecific hybrid via embryo rescue. If it doesn't work because your examples may less suitable for that. Yes, there are bridge varieties or bridge species, but I don't know by heart which is that. Once I will, I need to go through all those presentation I created in the past to find this information.
Thank you.
I think Chilense. There is a. Why is there? Hello.
So it's your
I'm using only one phone.
We have echo.
Can you hear me now?
Yes.
Okay, there is no. Yeah. No, I was just wondering because, Dr. Martha Mushler, she has been using Chilency 712, LA 712 to transfer acyl sugar based insect resistance to tomatoes for the last almost like 40 years now. Although they have made a lot of progress because it is lying on chromosome 3, but the linkage drag has not given us anything that is commercially sizable. So I think the question will be very relevant that I also haven't seen a paper where a bridging species, I would say that probably a pimpinellifolium accession would be more easier to use first and then you transfer it to the larger fruited. Would that be possible? I'm just giving a conjecture. I will also have to study if there is a paper somewhere. If anybody else has seen a paper like that, it's a good question because it's really a very tough species to transfer genes into, because drought also, Chilency has a lot of drought, but nothing commercial, I think, as yet that I know of has happened. Thank you. Unless anybody has something relevant to this, what is the next question?
Yeah, can I ask the question?
Why?
Hello? Just stop the call. Hello. Yeah. I have a question. That is there any molecular markers for cucumber mosaic virus in tomato?
Not to my knowledge, but it doesn't mean it doesn't exist. At least, yeah.
Yeah, I also don't have.
It is not published. Maybe the company who put effort on CMV in tomato,
Yeah.
they develop. The other point, what I can add to the CMV issue, even the reliable resistance source is not discovered or not published yet. So first, yeah, we need to find the reliable source for the resistance.
The point which I asked the question is, in Maharashtra, in some places, both TMV and CMV is coming in tomato, both the
Yeah.
virus. So it gives the appearance of, they call it as a plastic virus. So TMV, we have a molecular markers, but CMV, I found out, I have searched so many literatures, I didn't get the information about the molecular markers.
Yeah, it's
That was my question.
not published as yet. Not published as yet. Sundarraj.
Okay.
Okay. Next,
Okay.
I think Saurabh Tripathi.
Yeah, hi. Good morning, good afternoon. Everyone. Thank you so much for such a nice presentation.
I think
Mr. Vishwajit,
I think Vishwajit has to be moved.
Yes, sir.
Can everybody mute? Please mute yourself and only can speak.
Yes, sir. I am in.
Okay.
I live in West Bengal. Actually, there is most difficult problem is heat test. In the month of February 15, the most probably I have to see the physiological disorder of tomato. So, what is the solution of heat test breeding of tomato?
Sorry, the line is breaking up.
Heat test. Most heat test in West Bengal, there is very heat test in the month of February. So the most probably I have to see the physiological disorder and heat trace, and what is the breeding procedure in heat trace?
What kind of disorder? Because heat stress can cause symptoms.
Blue zoom end and dropping in maturity stage.
Calcium deficiency. Yeah, that's one of the physiological disorders of it is also linked with humidity.
Aparna, why do we have today?
There is, at least to my knowledge, there is no easy solution for blossom end rot. You need to screen your material in a hot spot and see the reaction of your plant. It's most probably it's a complex trait, so I'm pretty sure we are not going to find a single gene or two genes which may solve this problem. Also, what I learned that the more elongated the fruits of your material, it's more susceptible to the blossom end rot. But in general, what we do in tomato breeding, at least most of the breeders who face the blossom end rot problem, to screen the material in hotspot and get rid of the susceptible or extremely susceptible material.
Yeah, I think you need to do more screening of wider germplasm. Yeah.
Yes.
Yeah, thank you, Aparna. Yeah, thank you, everyone. So, sir, my question is not specific to tomato, but in general for vegetable breeding. So, the first question is related to how the grouping, heterotic grouping of the germplasm and developing the line by restricting the crossing between within the heterotic group, especially in vegetable crop. Does it work well, considering that the traits what we are majorly focusing are like quality and disease trait? That is my first question. The second question is specific to resistance breeding. Like when we are specifically breeding for resistance, there might be some byproducts or some of the selections we might be getting not good for resistance, but for other agronomic traits. So in your practical experience, how you dealt with those selections and whether it turned out to be a good selections for you in your experience.
Yes.
So these are the two questions what I had.
Okay, let me start with the second question because it's easier and in fact it's a very nice example. When I started the tomato breeding, the first disease where I felt that my germplasm is weak was the late blight. So I started to develop late blight resistant lines, crossing the donor, the pimpinellifolium material L3708. And at the end of the process, beside the resistant lines, I received some lines with improved fruit quality. And those additional improvements came from the pimpinellifolium. So pimpinellifolium is let's say, the only white tomato species which has red fruit. And of course, the size is very small, so you need to do several top cross, back cross with the elite germplas. But at the end of the process, it was not the intention of this project, but we end up with some lines with increased fruit quality, extremely deep red color, extremely shiny, and extremely firm. So that was let's say, a nice surprise. Your first question was about heterotic pools, and would you Would you repeat again? I cannot recall the whole question.
No, like, is there a
Yeah, sure.
heterotic grouping, just like it is done in maize? Are there heterotic pools in tomato or other vegetables?
Not of course heterotic pool. More if you look at more and more breeding program in tomato, the breeders try to establish the heterotic pool, and also other vegetables. Of course, it's less easy or less straightforward compared to corn, for example, because they started 100 years ago, and the other difficulties with vegetables, or let's say complexity of using heterotic pool in vegetables, that if you look at corn, I don't want to hurt anybody, but corn is a relatively simple crop. So its yield, it's yield, if you, the trait, what you need to target, its yield, some resistances, standing ability or lodging resistance, and the water content at the end of the season. And if you see tomato, pepper, or cucumber, you have many quality traits which you need to follow and consider. So if you have the heterotic pools and you create your hybrids between the heterotic pools, of course, it's evident that you have higher chance for better heterosis at hybrid level. But in vegetable, beside the yield, you need to ensure or secure the quality aspects as well. So definitely, I would suggest to start into the direction, if you haven't started yet in the breeding program, to establish heterotic pools and just follow the breeding scheme, what is related to that. But be prepared that it will not solve all your problem, and don't consider it as the only and universal magic approach.
Yeah, actually, I would like to add, Joseph. I mean, it's in vegetables, tomato, the kind of quality traits that we need. We don't find that more in the field crops or row crops. So here, just a little shape varies, and farmer will not take it, consumer will not take it. So I think, but on a inheritance basis, like I have done some work on tomato, you can, if you want, but you need to have a very diverse germplasm base. You should keep more fruit number and smaller fruit weight, and either indeterminate or semi-determinate as your determinates as your parents, male parents. On the female side, if you keep larger fruit size, less fruit number per plant or less number of fruits per cluster, the F1 will result into a heterotic hybrid for yield. But unless you have all the traits you need for your F1 hybrid in those lines, you will not be able to get that, like you can get in the maize. But I have also talked about it in my talk in the tomato symposium. I think I have a nice slide on that. I can share that later with Aparna. She can share it with the people here. But to do heterosis here, it's like same thing happening in rice. We have not been able to get real good heterosis because we get heterosis for yield, but then the grain is of no quality. That cannot be sold. We have not reached the millions of hectares that we wanted to do in rice, even after 30 years of working. Because we are working it like a grain crop, we need to first diversify for each of the agronomic traits within the quality trait. Then only you will get the heterotic pools you want. So that's my little addition to.
Yes. Yeah. So as I said, the approach is right. So,
yeah.
so
it's a long. I was recently in IRI, and that's what the Basmati breeder said, that finally after RH10, when they did the first Basmati hybrid, they stopped. They have now diversified the male and the female pools in Basmati, and now they are making their first hybrids with that, and they are starting to see success. So it's a long-term approach that you will have to do in vegetables. Yeah, anybody else? Aparna, do we have time? Oh, yeah. Makram Pandey is our tomato breeder. Makram, how are you?
Thank you.
Yes, please.
I'm doing good, sir.
Yeah,
Thank you. Nice to see you. And thank you, Joseph,
Good to see you. Yeah.
for such a nice and informative presentation.
Thank you.
Because this is resistance breeding, so I think every resistant variety has one or the other trade-off. So I just wanted to know how you being in R&D, you might develop a very good resistant variety, but it lacks in one or the other fruit trait or yield. So it's very difficult to sell to the commercial team because of some kind of trade-off. So how you deal in Syngenta these situations? Like you have very leaf color. So I am curious to know.
Yes. Just to make clear, I'm not dealing at Syngenta anymore. But let me share my my experience. Sales and commercial team and marketing team, they are eager to ask for all kind of resistance what they just found. And if they are not aware of the trade-off, they think that, okay, we have a nice variety. If I add TYLCV resistant on the top of that, my variety will be more valuable. That's true under certain conditions. If the pressure is there, the TYLCV pressure. We experienced a few times when commercial team was pushing for conversion of existing varieties to adding TYLCV. Let's stay with this trait. But because they said, oh, time to time we have a slight infection. I don't want to discover which area, which market it happened. Time to time we have slight, so the grower would feel more secure if we can sell them the TYLCV resistant variety. But it has not been communicated clearly that the resistance has a trade-off of 5% less yield. So when they tested the converted variety on the field and the growers had seen that, I said, okay, nice, but it's resistance. But you see the susceptible crop next to it looks exactly the same, but better yield. So if I have a very slight infection of TYLCV, so every 5 years I lose 5% of my plants due to the virus infection, I will not take a variety which every year will yield 5% less. So this is how to say the way how to manage that, because if you have every year serious problem with TYLCV, so if and you have the solution which deliver 5% less year, but without losing any plants or any yield due to the virus infection, and the susceptible variety has yield loss 10, 15, 20%, of course, growers will accept this trade-off, because at the end they will harvest more from the resistant variety. So my message is that, okay, if we have the trade-off linked with the resistance, you need to be very conscious and very aware where to use it, and only in case of really need you need to use it. Otherwise, yeah, just to create a fancy variety is
exactly.
Is not.
Makran, this is a classic, breeders and marketing conflict, because they want everything in every variety and tomorrow, and that doesn't happen. Breeding takes time, and they will always say, only this type I want. Slight change in shape, they cannot accept. So it's a unanswerable question, I would say. But I think your marketing people had to understand the trade-off. I hope you can make them understand. You can't build everything for everywhere with one hybrid. You need to choose different hybrids for different places. Deployment has to be different.
Thank you.
Okay, can we take
Yeah, thank you, Aparna. Thank you, Tikoo sir, and then Joseph. Of course, I had a good opportunity to work in vegetable crops, particularly tomato, and also with Dr. Tikoo. My kind of it is not a question, but per se, based on your both experience, now we are approaching a situation where dynamics, distribution, diversity of pathogens is quite vulnerable in across temperate and tropical climate, and the fluctuation in the weather, fluctuation in the pathogen is quite diverse. And we see limitations of host genetics in terms of the availability of durable resistance source. As a breeding expert, what is your foresight for the next coming years? Where do we land in? Do we have enough compatible resistance sources? Are we really making a breakthrough in terms of stable resistances? How can we approach the future trend of climate change, pathogen diversity distribution, both in temperate and tropical climate? Because tropical climate, every day things are changing, agronomic practices are changing, and not consistent follow-up. What is your future suggestions?
Very relevant point and very relevant subject. I can assure you that not only tropical zone is suffering from the climate change, even in Hungary, in temperate and continental climate zone, we face every year very strange weather behavior. Yes, this is the point. What you raised is really, I would say, the one of the most important points tomato breeders and breeders in general has to consider, that we need to be prepared that the situation is getting worse and worse in terms of disease pressure and in terms of combination of the abiotic stress and biotic stress. And that's why in my presentation at the end, I was calling to take action and really let's say take a kind of message to the decision makers that governments needs to be aware of that and needs to support scientists and breeders to tackle with this problem. Also, we need to unify our efforts and knowledge and expertise, because yeah, if you see the challenges, yeah, it's increasing every day, and if we just trying to solve it in our own corner, it's much more difficult than if we come together and say, okay, let's put together what we have in Japan's in expertise.
Yeah, one of the key.
I know that this collaboration is very tricky.
But, yeah, the key concern is
trust. But I would really love to see that happen.
Yeah, Suresh,
the key concern of investment to R&D, both in the private public sector, is getting very limited. But there is a lot of requirement in terms of understanding the physiology, pathogen, and host is much required. Perhaps the decision makers, as you rightly said, and also as Tikoo mentioned, and then Makaran Pandey has also brought. I don't know how to build this kind of very vibrant discussions in the marketing team and investment of the capitalist and researchers. I think there is a huge gap.
It's challenging.
I have just one, because there is a similar question by Bapu Dev Kate. He says, thank you for such an excellent practical presentation. That's for you, Joseph. And his question is related to resistance breakdown, which is, also what Suresh and many others are saying. What happens in the evolution of pathogen? Or sometimes there can be strainal variations that can happen in the. So how do you think the required level of resistance can be maintained across? Would you like to say something on that, or?
You are asking me?
No, that was Bapu Devkate had the question, and I am asking Joseph. He says, how do you think the required level of resistance can be maintained across locations? Like in India, TLCV, it doesn't work everywhere. No? Some gene will work here, some gene combinations will work somewhere else. So is there a better method or something? Before I say
Yes.
something. Yeah.
I would say that, okay, we need to be aware of what we have in our hand. And also we need to be aware if what we have is sufficient to solve the problem, to tackle the problem in medium, at least medium, but preferably long term. If not, definitely we need to start from the beginning with trade discovery and scout for new resistance. The other aspect also to support the genetics with the right agronomics. That's another angle we need to investigate. But yeah, as we said, and I think everyone agrees who works in this field that the pressure is increasing. So definitely we need to invest more into the basic research and invest more into scouting for new resistance and applying more the advanced technologies like genomic selection, big data analysis to learn more about this stuff, and genome editing.
Yeah, I would like to add that, Joseph, that is the right approach actually, and I have felt for a long time, like if you take the original, I'll just give an example. If you take the original bacterial wilt resistant line called CRA 66 selection A, it came from French West Indies. You can put it anywhere, it will show resistance. But the material that generated out of that, it will show resistance in some places, other places it does not. So what is happening is because the trait is not governed by a single gene. It's complex. There are probably epistatic genes lying on different parts of the genome, which are also giving you that stable resistance of that original material. So what I feel is needed more and more is that we need to do our F2 screening across those hotspots, then select and then try to combine. Maybe we are able to get those epistatic genes pyramided together. I'm just giving it as one of the approaches, but we'll need more costs, more approaches, and I think this is where public-private combination should do, because this is at pre-compete level. They should try and find out whether we can get F3s and F4s that are resistant more across the hotspots, and then everybody uses that for their breeding to develop hybrids. Just as a thought, but I think I would like to close it here. There are many small questions. I think, Aparna, can you put it all together and maybe we can send this to Joseph, and then if we can get answers to them, we can also put it into a booklet, probably, because there are a lot of questions that need to be answered. I thank you, Joseph, for an excellent morning session. I hope it's not too late for your, no, I think for India, it's a little late.
It was a bit, it was early morning.
I hope similar session can be held on insect resistance in tomato, because a lot of insect resistant genes are known, but we are not able to take it forward.
Insect resistance is
maybe we should try and get somebody
even more heavy subject than.
Thank you very much. Thank you all to stay there. We still have more than 100 people staying, which shows how interesting was the lecture. We had 152 people at one time. So thank you all the attendees, and thank you, Joseph, again. Thank you, Aparna. Do you have to say anything in closing? Thanks.
Thank you.
Yes. Thank you.
Bye-bye.
Thank you all. Thank you, Joseph.
Okay. Thank you. Thank you. Bye bye.
Thank you, ma'am.
Hello, Kritika.
Hello.
Hello, are you doing PhD in vegetables?
Yes, sir.
Okay, okay. Madam Kritika, I wanted your email actually. Thank you.